Thank you for this lovely introduction. It's a pleasure and honor to meet each and every one of you here today to share what we are doing at the University of Guelph and also get some feedback and comments and suggestions by the end of our talk. Good things comes in small packages. So we are in 2016. By 2050, the world population is going to be about 9 billion. So as of now, it's about 7 billion. There is a need to feed 2 billion people in about 20, 30 years. At the same time, the cultivable land or the arable land is shrinking. We also have to take into account the calorie count in terms of balanced diet and balanced nutrition for each and every citizen of this particular globe. So there are market agencies, also World Health Organization and Food and Agricultural Organization have done an estimate and analysis, and they have found out that in 2012, the global population needs about 20 gigacalories of food. By 2050, we need about 40 gigacalories, which means we need to double the amount of calories that need to be fed to the world population. At the same time, in terms of vegetarian as well as the food animals, when we look at the distribution between beef, mutton, pork, poultry, and other egg-related sector, we could see that 82 million is about the production of the number of poultry-based animals is about 82 million in 2005, but it is going to shoot. It has to meet at least 181 million tons of meat from the poultry sector, like layers, turkeys, and other related birds by 2050. So it is an opportunity and also a challenge to be able to meet this particular statistics, particular growth. So to be able to increase the food by 70% and meat production to increase by 50%, livestock sector has to overcome a variety of challenges. Recently, World Health Organization has released the statistics. Based on their data, they found out that every year there is 3 new diseases occurs. So out of those 3 new diseases, 2 diseases come from the livestock sector. Livestock includes dairy, poultry, pork or swine, and other related sector. And of these, 75% of the disease has a potential for being zoonotic, which means they can possibly cause disease to the human. Right now, you might have heard the virus, Zika virus, in the news. Just 2 weeks ago, we had about from Indiana province, state of United States, there was H7N8 avian flu virus strain outbreak has happened. So that's a brand new strain that has never happened here in North America. So that's a type of example of the possibility of disease outbreak in North America. So to be able to meet the challenge and also overcome the possibility of controlling the disease diagnostics, there is a need for shift. The shift is basically, instead of shipping samples from the farm, from the barn, from the dairy, basically do the test right then and there at the barn itself, at the poultry itself, at the barn itself. So we have agreed, a variety of scientists across the globe has agreed that we have entered the 4th revolution in agriculture. So we have passed 3 revolutions. The first revolution is basically mechanization due to industrial revolution in United Kingdom and Europe that happened in the 18th century. The second revolution is green revolution, basically doubling the amount of the production of crop using fertilizers and pesticides and a variety of techniques and strategies. So that was between the period 1943 to 1970s. And in the third revolution, it was predominantly in the United States, genetic engineering or genome-based technologies since 1973. So I would say for the past 1 decade, and we are also foreseeing to enter into an era called precision agriculture, or PLF, precision livestock farming. So basically, how we can do things precisely to be able to protect the public as well as the food animal from the disease, at the same time double the food production by using a variety of tools and technologies. So from a crop side, I would say there is about 9% loss in whatever we grow here in North America. So we are a well-developed country with such an advanced technologies. So whenever we grow a crop, after a crop has been grown, if we can preserve and store the crop from the loss, making each and every wheat kernel count through an advanced technology, that is a precision agriculture towards crop production. The same concept for PLF, which is precision livestock farming. So with regards to Ontario poultry sector, the world poultry meat production is about 93 million tons per year approximately. So poultry accounts for 41% of meat consumption in North America alone because of the protein and the other benefits associated in consuming the meat and the egg and the associated products. Here in Ontario, the chicken industry contributes approximately $2.72 billion worth to the Canadian provincial economy, and also creates approximately 19,000 jobs through food manufacturing, processing, poultry sector, and so on. So a small epidemic, a small outbreak can cause a huge impact in the economy of the province. So here's just a funny thing, a joke. Hey mom, what's this bird flu? Don't all birds flu? Then the little chicks, they say, let's eat. So the mom is thinking they have to learn a lot. The other joke you can see, the first time one of them sneezes, cut the rope. The message here is basically, if there is an outbreak in a farm, a poultry farm, that has to be immediately quarantined before a single incident that can potentially spread. For example, about 45 million worth of birds were euthanized in United States just a year ago. So that's a huge volume of bird, and also it created a significant impact in the poultry sector and the economy. So the goal, what is the missing link here? Why the diseases occurs, but how do we go about preventing the disease? The overall approach of our bio-nano lab at the University of Guelph, and also a variety of industrial sector, is enabling the producers to move from reactive. After the disease occurs, they try to prevent it. So to move from preventive to proactive approach through a predictive technology, which means even before the disease occurs, using precision livestock farming, sensing technologies, and nanotechnologies, we can predict, because of the correlation of a variety of factors, there is a possibility of disease to occur. So we can definitely go ahead and prevent the disease occurrence. So enabling the producers from reactive to predictive through proactive technology-based approach is the goal. So avian influenza statistics, about 48 million turkeys and layers were affected from high pathogenic avian influenza, and they were euthanized last year. Since 2003, about the past 12 years, billions of dollars were lost in the world economy just because of one particular disease that affected the poultry sector. Preventing the spread of avian influenza is the best way to keep the disease under control. So in this case, prevention starts with early diagnosis. We want to find out even before the disease occurs and monitor the health of the birds. So when a disease occurs, avian influenza, what are the possible symptoms or the clinical signs of that the bird go through? So we get flu, we have running nose, sometimes our temperature goes higher. Similarly, birds have a variety of flu-like symptoms. There are two strains, high pathogen versus low pathogen. Low pathogen all the time occurs. It comes and goes. Farmers can easily find out the possible symptoms. But the focus here is on high pathogenic strain, because high pathogenic strain creates clear symptoms such as ruffled feathers, soft-shelled eggs, depression. The motility or the movement of the bird inside the barn is restricted, so the birds kind of shy from making plenty of movement. So that is a symptom. And also droopiness, loss of appetite. They don't take the volume of feed they are supposed to take, and the water consumption also drastically reduces over a period of time. And also obviously the sudden drop in egg production because of the change in the metabolism inside the system of the chicken bird. So again, the possibility of zoonotic or the disease transmission from a high pathogenic strain of a chicken bird to human being is very, very low in North America. But in other countries, it has happened. So that's why the focus is to prevent and identify, and even before it occurs, there is a need to protect the birds. But low pathogenic, it is easy to manage. The management strategies include basically changing the feeding strategy, adding supplements, and also enhancing the hygiene of the poultry barn, clean, clean, and clean. So right now, when a disease occurs, veterinarians, food safety inspectors, they walk inside the poultry barn and then they take sampling. So samples could be from the environmental area. They can take samples from the swab or the oral fluid, or sometimes they also take blood samples from the bird. So imagine if the farm is so far from the animal health testing laboratory, because after they take the sample, derive the sample has to be shipped to the animal laboratory to be able to test it. So in the lab, commonly used technique is Rt-Pcr. It's a real-time PCR technique, so which takes about minimum 6 to 8 hours to identify whether it is a low pathogen or a high pathogen. But to be able to precisely find out what strain is causing in terms of H5N1 versus H7N9, the differentiation between the H versus N, it sometimes takes about 3 to 5 days. So that's a lot of time to be able to understand the result. So the challenge here is bringing down the time to results. If we can bring down the time to results and enable the farmers to know what is the strain that is causing the pathogen or the particular disease outbreak, and what is the subtype in terms of high pathogen, then effective management strategies can be easily employed to the disease prevention mechanism here. But the question naturally arises in terms of vaccines. So why don't we just go and provide vaccine to a variety of birds so that we can actually prevent the disease to occur. The major challenge is sometimes vaccines do not really work. Main reason is they are designed to induce HA in specific antibody response. So virus cannot survive itself inside the body. It needs a host cell. So it recognizes a cell inside the chicken system and make an attachment. So it is a survival mechanism to be able to flourish, prosper, divide, and then slowly spread through litter and also through other means. So hemagglutinin HA and neuraminidase NA glycoproteins, these are basically virus which has a tiny little coating, basically protein components on the surface of the virus, are the targets of the newly developed vaccines. There is also the availability of vaccines is very low, and because of the types, a variety of 16 types of hemagglutinin and also 9 subtypes of neuraminidase, causes a big challenge. So you multiply 16 by 9, it causes permutation and combination in terms of the division between H and N, causes a variety of vaccines to be developed. So by the time sometimes scientists joke, by the time they develop a vaccine for a specific strain, a new outbreak happens, that is H7N8 in this case, which happened just 2 weeks ago in Indiana. So due to this complexity, vaccine is still a challenge, and also because of cross-protective, cross-sensitivity related issues. So inactivated avian influenza vaccines are sometimes poorly immunogenic and needs high concentration of the protein. So you need a large amount of protein on the surface of the virus so that the vaccine can go and able to prevent that particular attachment to happen. So the virus doesn't spread, so you can suppress the proliferation of the virus inside the chicken system. So it's really difficult to predict avian influenza virus because it has the potential to cross the species barrier and potentially it can cause a pandemic. So ideally, yes, vaccine would be great because we can't, prevent the virus to attach to different host cells inside the system. But because of the practicality involved, vaccines may not be ideal, and early diagnosis just before the disease outbreak happen might be the appropriate way to go. So World Organization for Animal Health, they have said if one spill happens from the farm to the public, then it can cause a devastating effect. So Canadian Food Inspection Agency and the livestock industries are under enormous pressure to improve their biosecurity protocols and animal traceability through need of novel nanotechnologies. So that's how we come. So what we have done in our lab is we have developed a handheld smartphone appearing device. The device has the ability to detect the virus strains in just about 2 to 3 minutes, right then and there in the farm itself. So it could be commonly used by the poultry producer himself or herself. For example, if a poultry producer has a flu, and she is suspicious and she is not sure what is the reason behind this particular disease, whether it is a human-based virus is causing the flu or the virus has come from the poultry and it's causing the flu. To be able to differentiate between the human, like H1N1 versus H5N1, so there is a need to differentiate and discriminate and understand the preclinical screening mechanism using a nanotechnology based smartphone system. On the right hand side, you see a tiny little mechanism. So what we have done is we have used a semiconductor based nanomaterial. It is a quantum dot. So if there are two different glycan based quantum dot, glycan is the protein on the surface of the virus. So that particular protein is mixed with quantum dot in a functionalized manner along with an antibody. So in this particular mechanism, if a droplet of blood is being placed on the particular electrode, then both these three components come together. Because of the traveling association, when we shine a light, there is a change in the fluorescent intensity. So the tiny little change in the fluorescent intensity helps to recognize and identify which pathogen is causing that particular disease. So with respect to H5N1 versus H1N1, so it is H5 and H1. So the hemagglutinin differs, the protein composition differs. So the tiny little difference can be easily discriminated and identified, and the whole mechanism happens within just 2 minutes. So what is the beauty of this particular technique? So right now, when farmers derive samples from the bird, they derive milliliters of volume. So with the developed technology, all you need is just a droplet of blood. It's just like your home-based glucose test. Basically prick, take a droplet of blood, within a minute you know what is your diabetic level. The same concept, but here we can differentiate and discriminate the virus pathogen in a much more sophisticated and easier manner. It's very easy. Because of the nanotechnology involved, the high surface area to volume ratio, the amount of chemicals we are using in this particular method is very, very low. So unlike a lab-based test. So in the laboratory, you would need milliliters, sometimes macro scale volume of reagents and chemicals to be able to process and initiate the chemical reaction to be able to pinpoint what is causing and what are the subtypes of the particular components involved. So it is user-friendly technique. So the farmer has the ability to understand in a quantified manner as well as in a qualified manner. So if the farmer has to know whether it's a low pathogen versus high pathogen, they can go about quickly screening and differentiating using this device. We also went ahead and used an electrochemical technology rather than an optical technique to explore. Can we do multiplexing in terms of adding multiple varieties of H and N? Basically, all these H and N can be embedded in one particular device. So we would be able to use as a routine screening tool in the farm, no matter what particular strain happens. So with that concept in mind, we built another system here. What you see here, the yellow gold substrate on the screen printed electrode has the ability to initiate a chemical reaction. So again, it's very inexpensive. The idea is to bring down the test to $1 per test. So the cost decides the adoption from the farmer's perspective. So this is the device design you can see. It looks very similar. It also has the ability to transmit the data you collect in real-time fashion through an apps to a smartphone or a mobile phone, so that the veterinarians, the food safety inspectors, and the watchdog and the other agencies who oversee and supervises the disease pandemic outbreak can also obtain the particular record right away immediately. In addition, it also has added advantages of record keeping and tagging and labeling which farm is going through a pandemic with respect to a specific location and what kind of strategies or measures that need to be made. So we went ahead and did a variety of predictive technology strategy improvement. So this is a sensor which was built on a fabric using a accelerometer and an antenna. So it's the size of a dime or a dollar, very thin, and this particular 3-axis accelerometer and thermistor can be actually placed on the surface of the chicken bird. So it is more like a wearable device. So because of the thermistor involved, the thermistor would measure the temperature of the skin, and the accelerometer has the ability to monitor the movement and the activity. The tiny little antenna has the ability to transmit the data in a real-time fashion to the receiving smartphone. So you have the ability to measure the temperature as well as how much active the chicken bird is. In addition, there is a possibility to deploy a camera inside the cage or inside the poultry barn that acquire a live video recording of the chicken bird. So by correlating all these 3 parameters, we could come up with a way of identifying how healthy the chicken bird is. And we also did a variety of tests here. You can see the temperature drop. So the blue dot is basically represents the activity, and the red line here is the body temperature data. So it's infected with CKM-Z11. Those are not pandemic, those are very low. So you can see the drop in the temperature and the reduction in the activity level between a healthy chicken and a chicken that's being infected. So this data is indeed useful to be able to predict the disease outbreak even before it occurs. Here is another example that demonstrates the difference between the day and the night, and also the effect of the ventilation inside the poultry barn, how the airflow fluctuates that changes, the feeding time, the amount of water the bird takes. So all these parameters can be correlated and can be used as a mechanism to predict the disease. So we went ahead and developed a app for an Android-based smartphone system that would immediately relay the real-time data such as what is the temperature and relative humidity, light intensity, carbon dioxide, ammonia concentration, hydrogen sulfide, and so on. So why carbon dioxide and ammonia? So chicken take in oxygen and releases carbon dioxide. So the tiny little change in the concentration of the carbon dioxide is an indication in terms of telling what is going on with the metabolic rate of that particular bird. So it's one particular bird. If we have multiple carbon dioxide sensors deployed in a bigger poultry barn, we would be able to determine what is the tiny little change that is triggering and causing the disease causing markers in that particular schematic system. Here is an example of a monitoring system for laminated hen house based on Internet of Things. So it's not one particular parameter. We are going to measure the temperature, humidity, wind velocity inside the poultry barn. In addition, the skin surface of the chicken along with the activity using the accelerometer. So all these parameters can be correlated based on deployment. The question naturally arises, do I have to go and tag each and every chicken in a bigger barn or a farm? No, not necessarily. Using intelligent mathematical predictive modeling approach, you would be able to decide how to go about deploying the lowest number of sensors on the surface of the bird to enhance the mechanism towards understanding and collecting the necessitated data to be able to predict the disease. So it's a photograph that shows the deployment of the sensor in a barn and the topology of the transverse hen house sampling points that was decided based on mathematical formula. So again, being precision livestock farming, we are not interested in big changes. We are always interested in small changes. Instead of a clinical, we are interested in subclinical data points, because subclinical data points and the tiny little changes help us to actually predict and estimate when it is going to happen and what is the possibility or probability of occurring a disease. This is another example of remote monitoring of hen house experiment. So we can design based on integrated wireless sensor network using variety of electronic components. These are very inexpensive to be able to provide a low-cost solution to the producers and farmers. So the data we are collecting here is a self-decision estimate value with respect to temperature and the number of data points, and we can estimate the tiny little change in the carbon dioxide from a node or a network of sensor, which zone the particular carbon dioxide is dropping with respect to the density of the chicken bird inside the particular barn. If the density decreases or increases in terms of the population of the chicken bird, then we can correlate that with respect to the change in carbon dioxide concentration to be able to make an estimation in predicting the disease. So along with carbon dioxide gas sensors, that we could also deploy e-nose or electronic nose. These are basically volatile metabolite sensors. So based on the drooping, based on the manure and the poultry litter, traces of chemical components are being released inside the air of a hen environment. So the tiny little changes of that particular chemical, such as aldehyde or alcohol or ketone, those metabolite marker can be easily decided and captured and quantitated using sensor technologies. So they will be able to transmit in a real-time fashion to make a early warning system and a judgment. So a hybrid environmental and population density management system. I was reading the news just a week ago. Tim Hortons and Burger King, these are two companies, they have decided to go about obtaining eggs from non-caged poultry birds by the year 2020 from United States as well as in Mexico. So because of the awareness towards enhanced animal welfare, there is a need for a free range but in a protected environment to allow the bird to able to produce the egg in a naturalized manner. With that context, the system of poultry production may have specific impact in the Canadian sector. So with that context, we went about producing a low-cost technology by incorporating a variety of microelectronic components to be able to enable the users to predict the technology and the disease. So here is an example of the density management system. So you can see a chicken bird, they are trying to gather. In certain locations, they are scattered. Because it is a video, which is a composition of a number of frames of pictures, we could also determine the activity involved with respect to the feed. So we could also correlate how do they feel right after they have the feed, and what is the disease transmission mechanism from one bird to the other bird. So we can go about making a very precise transmission identification mechanism using a combination of these imaging as well as the volatile metabolite measurement technologies. So these are the screenshots of the components we were able to measure from the smartphone. We could see that temperature, humidity, the composition of carbon dioxide, as well as the density monitoring in a real-time fashion in a smartphone using the integrated electronic components in a real-time manner. So the graphical user interface and the adoption of the technology enable a real-time monitoring. The farmer can make a early warning decision system, early warning decision strategies based on the data they were able to collect. Here is another example. It is a switch to activate event-driven system for chicken health monitoring. So a MEMS device was developed. MEMS is basically a micro-electromechanical system. The idea behind this is if you watch the little kids who are wearing shoes, the shoes used to glow. The more they jump, the more they glow. So it is made of a material called piezoelectric material. Based on the motility and based on the movement of the bird, more amount of energy can be stored by the piezoelectric material, and that energy stored can be transmitted to the battery of the sensor that is attached to the surface of the chicken bird, just using a simple tape. So there is really no need of external power source. It will collect the activity of the chicken bird, how fast they move, and also using Doppler effect, you can go about measure the blood flow velocity. Again, it is a non-invasive. There is no pricking happening. It just sends a light signal, sound signal from the sensor to the tissue surface. Based on the Doppler, you are able to determine the frequency response and using a mathematical calculation, you can actually judge the change in the blood flow inside the chicken bird. So these are photographs that shows the size and the shape of the piezoelectric sensor that was developed. The sensor node can be wrapped around the wings of the chicken bird to be able to determine the measurement of the chicken during a specific duration. So again, with respect to change and the effect and the influence of light and the environmental parameters, what is the behavior with respect to change? So that can be determined as well using this particular sensing mechanism. So blood flow and physical activity data transmitted from the microelectromechanical systems and the piezo-based sensor can provide valuable data, including the activity as well as the environmental parameters along with the blood flow conditions. So you get a physiological data, you get a biochemical data, you also get the environmental data. So the next step is to how to make sense out of these data to be able to predict the diseases. So disease modeling and also software has to be developed to be able to come up with an approach to be predict with what much accuracy, with what probability the disease can potentially occur. So this is an interesting technology. The idea here is, can we do something in terms of measuring the sound of the chicken bird? So by collecting the sound of the chicken bird, because when the farmer enters the poultry farm, he hears first, he or she hears first, and then sees and then smells. So all these, so the ear, the nose, and the visual or the eyes, all these three can be replaced by sensors which are developed by nanotechnology. By using all these components, one could easily come up with a way to predict diseases even before the disease occurs. So what you see here on the screen is basically a signaled background processed frequency analysis of the amount of noise or the chicken talk measured using the sensors we have obtained. So the time frequency vocalization graph helps to identify how do they talk, or whether they have an antagonistic approach or a synergistic approach. We were able to find right after they feed, they were happy. So there is a change in the intensity of the noise they make. But just before they go and approach the feed, there is a difference in that particular intensity, and the signal to noise ratio also changes. So the tiny little peak, the tiny little changes can be attributed and correlated with the other parameters to be able to make an intelligent decision towards predicting the disease. So the goal is, because the how do we go about make an intelligent decision as a innovative detection and advanced diagnostic technologies for farmed animal health management towards smart agriculture and precision livestock farming. So biosensors would allow monitoring of animal health and well-being, and can also send text to alert farmers when an infection increases or even before the infection is going to occur at a herd level, as well as a decrease in the production that happens. So in an integrated farming system such as the one cartoon shown here, you have a poultry barn, you have a dairy as well as swine farm. So the collective integrated technology can help and aid the disease prevention in a much more intelligible fashion. Being a professor, I also teach a course called Bioinstrumentation Design. It's a popular course. As part of the course, students have an opportunity to experience building plenty of wireless sensor networks, and they also attempt to solve real-time practical problems in the lab. So by the end of the 12th or 13th week, they give a trade show presentation during which industries are invited to enhance the outreach and also to enhance the knowledge transfer. So this is a popular course. There are certain aspects of solving problems related to agriculture, livestock farming, and biomedical applications also happen in this particular trade show. Very quickly, Premier Kathleen Wynne, she declared and made a statement during October 13, challenged the Ontario agri-food industry to double the growth and also create about 120,000 jobs by 2020. So the technology we are involved in, the precision livestock farming, has the potential to create approximately 3,700 new highly skilled and technical jobs here in Ontario. It translates, the highly qualified personnel could be working on development of sensors and technology. A portion of the HQP might work in the barn or the poultry farm, and a portion may work in the food processing, data processing, as well as the software development related area. So mobile health technologies and apps will enable the alert and also can serve as an efficient biosecurity tool or a system for Canadian livestock sector. So with this, I would like to thank you and thank you for this opportunity to share what we have done in our lab.